fantastic trial, the DART trial, which is immunotherapy for rare cancers.
Thank you. I'd like to thank Dr. Kerdrack and the committee for inviting me to share my experience with DART, which is a national NCI SWOG platform trial for immunotherapy in rare cancers. Disclosures. So why do we use the NCI SWOG platform for rare tumors? Because the private sector, the pharmaceutical companies are not interested because it's a small market as an individual tumor. But as a group, it's not rare anymore. In fact, a quarter of all new cancer diagnoses belong to the rare cancer category, which is usually defined as less than 6 out of 100,000 population. And we know at the advent of the nivo-ipi regimen in melanoma, there weren't a good biomarker in this combinatorial therapy. Genomic, immune, PD-L1 wasn't great. So really a non-biomarker-based, histology-based basket trial designed solely for rare tumor was a high unmet need. And really want to replicate the success story of rare tumors responding to immunotherapy, such as Merkel cell or Hodgkin's lymphoma. So we designed DART, which is an acronym for Dual Anti-CTLA-4 and PD-1 blockade in rare tumors. I remember we first met, 3 of us at SWOG, Early Therapeutics and Rare Cancer Committee, and we thought this would be a great idea to do it together. This trial had a primary objective of response rate and applied the Simon's 2-stage design, 6+10. And we used the Checkmate 227 regimen, which is the 1 mg per kg IPI every 6 weeks, plus the Nivo 240 fixed dose every 2 weeks. So this is not the FDA-approved melanoma regimen, which has much higher toxicity rate. So how we began was, at that time, NCI already had a platform trial, NCI Match. This is a targeted therapy basket trial, and you have to have an actionable mutation to go into the trial. But it ended up being that about a quarter of all NCI Match patients belong to the rare cancer category. And a lot of them were screen failed because they didn't have the right actionable mutations. So NCI also had high unmet need. What do we do with these trials? There was a time when we proposed this trial, so we were able to, marry and get patients enrolled into DART trial when they screen failed from NCI Match and also belong to the rare cancer category. So we started planning for this trial year 2015. Took us like 2 years to get everything up and running. So activated the trial in January 2017. We started with 13 different cohorts in different baskets. And then now we have 53 cohorts. We enrolled last patients in March 2023. That was the last new enrollment. But then we still have active patients on the study, still ongoing. How crazy is that, right? So this trial has been open for more than 6 years, and with the help of the CMAC support, we were able to add the biomarker portion, the translational medicine piece to this trial as well. So this is our initial 33 cohort, and as you can see, we have different rare tumors, but also have histology agnostic, or the primary organ agnostic neuroendocrine carcinoma, which could be salivary gland, lung, breast, prostate. It could be anything. We did have tumor of unknown primary group. We did have not otherwise categorized group, NOC group, and we were accruing patients that I've never heard the name of during my oncology training. So it was exciting to see all these rare cancers being able to have the opportunity to participate in this national immunotherapy trial. So we added a few more and ended up in 53 different cohorts, the last cohort being the prostate neuroendocrine tumor, which is therapy emergent small cell. And these were picked up as we were doing trial, as we were seeing high unmet need. We just revised the protocol to expand on different cohorts. One example is we saw the New York Times article about young women with very rare cancer called small cell carcinoma of ovary hypercalcemic type, responding really well to immunotherapy. So we added that cohort. Per Dr. Kurdrak's suggestion, we added PD-L1 amplified cohort, the first biomarker-based cohort, pan-tumor cohort, by NGS, not IHC. We had a terrible confusion there, but this is a very innovative cohort that we were able to just add real time. And I think this is a major innovation that we were able to achieve by running this trial for a very long time. So this is our SWOG stat. We were able to real-time update the patient accrual status. On the right, you can see the sites that opened our trial. So there was a high unmet need. Most of our sites were community sites, and we were able to open the site in over more than 1,000 sites across the United States. We ended up accruing close to 800 patients over 6+ years. So the bottom line is this trial actually saved many lives. When we had stories, newspapers, one patient from Chicago had adrenal cortical carcinoid in his late 20s and got CR and still enjoys durable remission. It's my clinic, my patient had esophageal neuroendocrine tumor, another durable PR, metabolic CR. We got letters from patients telling us their experience, how grateful they are because of this trial being made available for them. This was an award that we gave to our patient during our ceremony. So the overall result is that out of 727 eligible patients that were actually treated on the regimen, almost half of all cohorts, so 25 out of 53 cohorts demonstrated clinical activity as defined as at least 2 patients with confirmed radiographic response. The median objective response rate was 12. There was no new AE signal. We already know that grade 3 AE is about 20% in the nivo-ipi regimen. So by cohort, you can see that neuroendocrine tumor had objective response rate of 25%, 1 CR, 7 PR. If you only look at the high grade, it goes up to like 44%. Sarcomatoid lung was 29%, angiosarcoma 25%, metaplastic breast cancer 12%. You can see many other different tumor types that you really would not have thought would show CRs and PRs because we traditionally thought those were cold tumors, not able to respond to immunotherapy. And what was interesting is our statistician, Megan, looked at the response rate of tumors, and she broke down the granular level and looked at the actual change in tumor size by RECIST and how that correlated really well with the 1-year overall survival rate. It almost looks like a linear curve right there. So this was published JCO. And then the other thing we found is that 6-month PFS across all different cohorts correlated really well with the 1-year and 3-year overall survival in our data. So, and then the amazing thing is the most responses were lasting more than 2 years. Many are still ongoing responses, which we really didn't expect when we began our trial. And among those patients with CR, PR, or PFS at least over 2 years, these are 76 patients still doing really well. And you can see the arrows of ongoing response patients. And first, when I looked at this graph, the x-axis, I was thinking, is this, because for like phase 1 trials, you would think that x-axis was really like 2 months, 4 months, 6 months, but this is 2 years, 4 years, 6 years, and 8 years in the y-axis, and you see this kind of tumors plot in this basket trial of rare cancers. I think this is just mind-blowing to see that so many people have responded to this regimen. So to go cohort by cohort, neuroendocrine tumor with high grade, you can see that there is a deep durable response seen, which led to the incorporation of the nivo-ipi regimen in the NCCN guideline. We have angiosarcoma. Again, whether it's UV associated or radiation associated, you can see that there's a durable, deep response, exemplified by the facial angiosarcoma responding really well to the regimen. And also, the sarcoma committee has incorporated our regimen in the angiosarcoma NCCN guideline. We have metaplastic breast cancer data showing 3 patients had very deep, durable responses. We are doing the translational medicine piece to look why, but it's encouraging to see that there are these patients who are responding for a very long time. 3 of them still ongoing at this point. We have bronchoalveolar cancer, which is rare. Thyroid cancer, these are well differentiated mostly, and they still, when we looked at them, they're still cold tumors. There's something about CTLA for bringing in the lymphocytes into the cold microenvironment, and they still respond. Salivary gland, non-adenoid cystic, you can see there are a number of durable deep responders there. Vulvar cancer, small cell ovarian cancer, I told you about 2 deep responders. Clear cell ovarian cancer, there's a lot of them that responded. And granulosa cell tumor, which we traditionally thought was really just hormone-driven tumor, but we did see a nice deep response in few patients as well. And PCOMA, which is a super rare tumor, we got a few responders there. Pancreatic neuroendocrine neoplasm as well, and the gallbladder cancer as well. These are MSI high, not non-MSI high tumors. Vaginal cancer, we actually have a poster being presented at this AACR conference for our vaginal cohort. So we're still actively presenting our cohort data. Small bowel cancer, these are non-MSI high cancers that really still showed a response in two PRs there. Desmoid tumors, we actually had an oral presentation last year for desmoid tumor result, but when everything is resolved, these are not like treatment naive. These are patients who exhausted all different treatment options, including surgical options, obviously. Sarcomatoid lung cancer, obviously you see that 4 patients had PR. Adrenocortical carcinomas also had many deep, durable responders. And we also had rare tumor gestational trophoblastic. So 3 out of 4, a very small cohort, but objective response of 75%. And these tumors, the response is being evaluated by the beta HCG level. So you can see very nice chemical response here, and a lot of them really did well in terms of overall survival. And all these data are publicly available, published for your information. So another interesting observation from this trial is that patients who discontinued the therapy in the first 6 months of the treatment because of the immune toxicity had longer overall survival than those who discontinued treatment for other reasons other than the immune toxicity. So you can see it nicely in our cohort of 800 patients. So now we know there are responders, but what can we learn from this trial? Obviously, translational piece, I think, is the vital, essential piece of this trial, and that is what I view as a patient's legacy that we need to learn from. So we are looking at genomic immune landscape of these rare tumors. What are the signature for responders, resistance, and also immune-related toxicities? And that is across really different cohorts. So we've partnered with NCI Match, partnered with CMAC. So we have tissue pre-treatment, we have blood at 3 different time points, baseline, first imaging, and progression of disease. So these are our collaborators, commercial and CMAC based, and we have CMAC and CIDC collaborating on different assays. We have 3 different tiered assays that are part of the CMAC CIDC, and these are the team working on various projects of the translational medicine piece. We started our translational research with the neuroendocrine piece, and applied whole exome sequencing, PD-L1 IHC, and multiplex IF. Also, we looked at the cytokine, chemokine, CyTOF, and TCR repertoire examination. And some of the preliminary findings that I can show is in this cohort, we didn't really see PD-L1 correlating with the response. However, we saw that B cell or dendritic cell signature from the tissue correlating with the response. And the tertiary lymphoid structure also seemed to be correlating with response in this neuroendocrine cohort. Immune cell infiltration didn't really change by grade of neuroendocrine tumor. And we did see using the O-link that combinatorial therapy promotes the immune mobilization. With CyTOF, we saw discrete effector memory T cell subsets that differentiate response at baseline and early on treatment. And when we looked at the thyroid cohort, also we didn't see the benefit of looking at benefit of PD-L1 or the T cell infiltration correlating with response. But what we found the double positive PD-1 and PD-L1 double positive TILs did correlate with response. So we're very preliminary, but looking into what that means. TMB did not correlate with response, at least in the thyroid. So I'm just giving you an example, but you can imagine this is an extensive effort to look at all these cohorts at each cohort level, but also across tumor level. So we're trying to build, because we got the second funding approval for the CMAC, so we're going to really look into these rare cancer tissue and build like rare cancer TCA out of this project. So really, we're going to delve into the next questions using mass spec to look at the immune relations to immune cytotoxicity, radiomics, pathomics using the H&E slides. We're scanning them as we speak. Also definitely looking at phenomics and really this multi-layered information. We are hoping to use AI model to look into that. So we're using the mass spec technology, as you can see here. And my group has preliminary been showing that, yes, this serum proteomics can help us to look into the immune-related toxicity. So we're hoping to replicate that in the DART cohort. So the take-home message is that, yes, we can accrue to these rare cancer trials. It's a platform trial. We found there's really high unmet need. Still, a lot of rare cancer patients have no access to clinical trials that utilize immunotherapy. And it's not like, well, we tried few of them and they didn't work. No, we found so many deep, durable responses across different, more than half of the cohorts. And we are learning because we have stored their tissue and blood. So there's still high unmet need, so we're still pushing hard to make this a continuum of platform trial. So we didn't give up preparing for the DART 2. So the platform can be there, and then we can switch different agents, just like the iSpy or iMatch. The platform trial for rare tumor is definitely a high unmet need, but we do have a strong success story from the DART 1. Yes, we are learning from the multi-omics translational medicine efforts from the DART tissue and blood samples. So really the team science from the CIDC, CMAC, SWOG team is currently in action. So stay tuned for making the portraits of care TCGA immune landscape, the results. So we remain excited. So the last part is for personalization of combination immunotherapy, AI-based comprehensive biomarker discovery and modeling can truly be applied. And we're using really the multi-layered data points, proteomics, radiomics, pathomics, and we annotated all these precious clinical outcomes in a very accurate fashion. So we're very excited to do that as we speak. So I'd like to thank everyone been involved in this tremendous communal effort. I'd like to thank my colleagues, Dr. Kurdrak and Dr. Patel, who are the study chairs of this amazing trial. And I'd like to thank Dr. Blanky, Dr. Ryan, Dr. Othis, our main statistician, and Dr. Wistuba and Dr. Haymaker, who are our CMAC lead to do our study. And I'd like to thank BMS for supplying the drug for us to do the trial. I'd like to thank Marcia Horn, who's our patient advocate for our committee, as well as the DART trial. And really like to thank everyone involved, and especially for patients and their caregivers who made all this possible. Thank you.
So if there's questions, Dr. Young-Chae, that was wonderful, and we're happy to address questions. And in the meantime, I have a question for you. So do you think after doing this, it would have been better to do the biomarkers up front or to do them afterwards? I'm always a biomarker up front person, but DART made me think that doing them afterwards was also okay. But how do you think that balances out?
Oh, for this trial, we didn't have the funding. We got the CMAQ funding, so we were able to add it later, but that was just how it panned out. But I agree that we should bank every patient's tissue and blood up front. But whether we're using any companion diagnostic to select patients, I think at the start of the trial, and maybe we still don't, have enough evidence to say, only this group will work. So, it's really just as we do, as we learn, kind of go as we learn trial. So it is still an exploratory trial, but based on this, hopefully we can go into a future biomarker-based trial. And one of the ideas I had, and which I presented, is the TIL-based trial, the tumor. We have MSI high, TMB high. What's next? It's the tumor infiltrating lymphocyte high. But how do you measure that? Most accurate way, most practical way. So, that could be a biomarker-driven or companion diagnostic led trial. But trial like this for rare tumors, I think we're still in a phase of learning. So, yes, thank you.
I have a comment. May I?
Yeah.
So I practice in a very open environment where we have nothing, almost 80% of our cancer therapies are not covered by insurance, and we don't have insurance. So everything is out of pocket. And this is a comment and a compliment. Great presentation, great trial. Me under tremendous pressure now, because every patient has to get Nivo-EP now. I can't let a patient go without seeing Nivo-EP. I mean, the study is so remarkable. It just drives home a message wherein my message for the day is for all these rare cancers where we really don't have a solution, there may be one. And so if a patient comes and says, do everything for me, I'm compelled to think Nivo-EP after looking at the presentation. So congratulations, great work.
So,
Thank you.
did you have a question?
Yes. Hi. Thank you so much for your very insightful presentation. So, and maybe my questions might be naive. So the rare cancers depends on the frequency that they are diagnosed. Are you mostly interested in those that show high aggressiveness? Because of course I saw in your endocrine prostate carcinoma that it's a very rare entity, but still it's a lethal. And then the next question is like, for the patients that you decide to treat, are you treating them at the primary setting, so when they are diagnosed, or you're waiting to get metastasized to decide if you're gonna perform any type of treatment? Thank you.
Thank you. Good question. So the first, we don't discriminate based on their aggressiveness. So one example is adenoid cystic carcinoma. A lot of them are known to grow very slowly, but in the end, people die of disease as they don't respond to chemo. Actually, there's no good IV systemic therapy that works well. I mean, there are drugs that will likely be approved, but they have to exhaust the standard of care options to be able to come onto the trial. So they have proven refractoriness to the standard of care, or the standard of care has not, it's approved, it's available, but sometimes it has never really been shown to effectively increase overall survival, So yes, they have to use standard care, and whether they're aggressive or not, just like in case of adenoid cystic carcinoma, actually we saw responder even in adenoid cystic carcinoma. Yeah, so those are good examples that we can enroll to any rare cancers with different growth speed.
Well, thank you very much. That was a great presentation. And Dr. Lamai is now going to present the view from India. I'll just make, as she's getting up, a brief comment that all these patients had failed standard of care, at least one.